Good morning each and everyone. My name is KJ Lausa, Bachelor of Science in Agriculture, major in Animal Science 4. Today, we will be talking about environment application bioremediation. So, what is bioremediation? It says is a collective phenomenon involving processes that use biological systems to either restore or clean up contaminated sites. The microbial community is consistently reported for bioremediation. Most of the indigenous microbes have the ability to successfully bring up the environmental restoration via oxidizing, immobilizing, or transforming the contaminants. It aims to reduce or bring down pollutant levels up to a detectable, non-toxic, or acceptable. The concept of bioremediation was first used on a large scale in 1972 for the cleaning of sun oil pipeline spill at Ambler, Pennsylvania. However, in laboratory scale, George M. Robinson was the first to recognize this process during experiments on sewage and oil treatment. Subsequently, in 1992, Environmental Protection Agency or EPA developed protocols for bioremediation on the process of different case studies on bioremediation. The modern approaches of bioremediation are to search for a novel microorganism from contaminated sites. The isolated microbes are thought to have a strong potential to remediate pollutants. The use of genetically modified strains and also microbial consortium has been used directly or indirectly to increase the bioactivity of the remediant. Various mechanisms including bioaccumulation, biodegradation, pathways, and different modes for bioabsorption have also been investigated for the removal of pollutants. In this chapter, we highlight and discuss different bioremediation processes and their classification with future potential. It says also, bioremediation is an emerging technology which can be simultaneously used with other physical and chemical treatment methods for complete management of diverse groups for environmental pollutants. It seems as a sustainable approach for the environmental pollution management, and hence there is a need for more research in this area. Efforts need to be made to generate a synergistic interaction between the environmental impact on fate and behavior of environmental contaminants and assortment and performance the most suitable bioremediation technique and other relevant technique that can sustain the effective and successful operation and monitoring. Vermi biofiltration and genetic engineering technology can be promoted and adopted at larger scales for sustainable waste recycling, polluted soil treatment, management of solid waste with certain optimized conditions. The continuous efforts of research and development will direct future regulations dealing with bioremediation targets, contaminant availability, and their potential threat on natural ecosystem. Moreover, the availability and biodegradation of contaminants in any natural or man-made system and degree of threat to human health caused by various environmental pollutants would be more easily forecasted by using multidisciplinary technologies. There are three types of bioremediation. First, biostimulation. Second, bioaugmentation. And third, intrinsic bioremediation. Bioremediation has a triple corners process. These are pollutants, environments, and organisms. Under pollutants, we have two elements. These are organic and inorganic. We'll note that organic has three composed elements. These are solid, liquid, and gas. Environments compose also soil, water, and air, and organism has also microorganisms, plants, and enzymes. Redox cleanup reactions. Anaerobic or aerobic metabolism involve oxidation and reduction reactions of redox reactions for detoxification. Oxygen could be reduced to water and oxidized organic compounds. Anaerobic reaction can use nitrate. In return, biomass is gained for bacterial or fungal growth. In many cases, combined efforts are needed. Indigenous microbes found naturally and polluted sites are useful. Bioremediation uses also a bacteria, greatly affected by unstable climatic and environmental factors from moisture to temperature. For example, pH in soil is slightly acidic. Petroleum hydrocarbon degrading bacteria do not work well less than 10 degrees Celsius. These microbes are usually thermophilic and aerobic. Fertilizers are needed. Seeding or bioaugmentation could be useful too. These contain monooxygenases and dehydrogenases to break down organic matters, including most toxic substances. Use of fungi in bioremediation. Yes, bioremediation uses also fungi. Candida can degrade formaldehyde. J. beberella can degrade cyanide. Slurry phase bioremediation is useful too, but only for small amounts of contaminated soil. Composting can be used to degrade household waste. White rat fungi. White rat fungi can degrade organic pollutants in soil and effluent and decolorize craft black liquor. Example of this is PC or Phanerochaete chrysosporium. Can produce aromatic mixtures with its linolytic system. Environmental cleanup process. The basis of removal and transportation of waste for treatment. Basically, there are two methods. First, in situ bioremediation and ex situ bioremediation. So what is in situ bioremediation? Meaning, it involves direct approach for the microbial degradation of xenobiotics at the sites of pollutants. Example of this, soil and groundwater. It has been successfully applied for cleanup oil, spillage, or beaches, etc. There are two types of in situ bioremediation. First, intrinsic bioremediation and engineered bioremediation. Ex situ bioremediation. So, the meaning of this is the waste or toxic materials can be collected from the polluted sites, and the bioremediation with the requisite microorganisms can be carried out at designed places. Bioremediation of hydrocarbons. It says petroleum and its products are hydrocarbons. Oil constitute a variety of hydrocarbons. Examples, silenes, naphthalines, octanes, camphor, etc. The pollutants can be degraded by a consortium of microorganisms. Examples, Pseudomonas, Cyanobacterium, Arthrobacter, Microbacterium, and Nocardia. Genetically engineered bacterial strains are used to enhance bioremediation. In 1979, Anand Mohan Chakrabarty he obtained a strain of Pseudomonas poteda that contained the XYL and NEH plasmid, as well as hybrid plasmid derived by recombinating parts of CEM and OCT. In 1990, the USA government allowed him to use this super bag for cleaning up of an oil spill in water of state of Texas. It was produced on a large scale in laboratory, mixed with straw and dried. Example of the oil spill in USA: marine pollution causes sewage from cities, industrial effluents, oil slick, pollution from warship, and military activities. Oil spill: 11 million gallon oil spill from the supertanker Exxon Valdez, Alaska on 1989. Straw was sprayed over oil slicks. The straw soaked up the oil and Bacteria broke up the oil into non-polluting and harmless products, as you can see in the illustration on the screen. But phytoremediation is use of plants for accumulation, removal, or conversion of pollutants. So what is phytoremediation? It says effective and low cost, environmental friendly, soil cleanup of heavy metals and organic compounds. It says the advantages of phytoremediation. First, effective and low cost, environmental friendly, soil cleanup of heavy metals and organic compounds. Second, pollutants are absorbed in roots, thus plants removed could be disposed or burned. Sunflower plants were used to remove cesium and strontium from plants at the Chernobyl nuclear power plant. Transgenic plants with exogenous methylthionine or a metal binding protein used to remove metals. Bioremediation of metals contaminated environment. I will be giving to you the illustration on the screen. What is biosurfactants? Biosurfactants are chemically compounds characterized by hydrophobic and hydrophilic or non-polar and polar regions in one molecule or amphipathic molecules. Biosurfactants from bacteria, cyanobacteria, fungi, and yeast are classified into 5. There are glycolipids, lipopeptides, phospholipids, glycoproteins, polymeric biosurfactants. Biosurfactants producing gene, genetically engineered Pseudomonas aeruginosa. This new strain can produce a glycolipid emulsifier. It can reduce the surface tension of an oil-water interface. The reduced interfacial tension promotes biodegradation of oils. Bioremediation of industrial waste. A variety of pollutants are discharged in the environment from a large number of industrial and mills. Bioremediation of dyes. Second, bioremediation in paper and pulp industry. So now we will be talking about the prevention and control of pollution. These activities are supported by set of legislative and regulatory promotional measures such as policy statement on abatement of pollution in 1992 at the National Environment Policy 2006. The major actions in abatement of pollution and environmental cleanup are as follows: clean technology, control of pollution, environment research, forest protection, hazardous substances, clean development mechanism. There is an immediate necessity for initiating an all-India coordinated project on bioremediation of the contaminated sites, involving premier institutions in this field for conducting large-scale demonstration projects. Furthermore, various departmental agencies and related ministries in India may plan a joint action plan to launch bioremediation from lab pilot. Bioremediation from lab pilot field scale, as you can see on the screen. We proceed to advantages and disadvantages of bioremediation. So, advantages of bioremediation. First, bioremediation is a natural process and is therefore perceived by the public. So, it can easily perceived by the public. And it is a natural process, not an artificial processes. Second, bioremediation is useful for the complete destruction of a wide variety of contaminants. Instead of transferring contaminants from the environmental medium to another, for example, from land to water or air, the complete destruction of target pollutants is possible. Bioremediation can often be carried out on site, often without causing a major disruption of normal activities. Bioremediation can prove less expensive than other technologies that are used for cleanup of hazardous waste. So those are the advantages of bioremediation. Now let's proceed to disadvantages. These are some concerns that the products of biodegradation may be more persistent or toxic than the parent compound. Biological processes are often highly specific, microbial populations, suitable environment growth conditions, and appropriate levels of nutrients and contaminants. It is difficult to extrapolate or deduce from bench and pilot scale studies to full-scale field operations. Bioremediation often takes longer than other treatment options. For references, bioremediation is its application to contaminated. You can see at this link on the screen. So thank you. That's all.